or so, we were trying to establish the science of it before we roll out to the school. Now, the second part is transitioning into science education. So that's a part where we try to translate translational science, so to speak. Translate the science research at the school level. And then the third part of this journey has to do with the proof of concept, the use of citizen science, involving not just a student, but also government agency, so that it makes the project a little bit more authentic to the students. So this part of my talk, there'll be three subparts. I'll talk about the science, I'll talk about the translation, and then I'll talk about citizen science. Next slide, please. The science. Ecotoxicology, what in the world? What do you mean by ecotoxicology? You might understand the word toxicology, but what is ecotoxicology? This is a field that my collaborator, Dr. Beverly Goh, is an expert in. She is a marine ecotoxicologist, all right? So this part of the talk is more about her than me. So ecotoxicology, what is the definition of ecotoxicology? A brief definition of it is it's an integrated study of toxic effects caused by pollutants on ecosystems and biota. That's the overall, a very general definition of ecotoxicology. So basically, we are trying to understand the effect of pollutants on living things, all right? So what are these pollutants? So pollutants can be many forms. The traditional definition of pollutants would be they're usually of chemical of origin or synthetic. But more increasingly, they also include any types of environmental stress that's caused by humans. It could be heat stress, etc. It doesn't have to be chemical. It could be a physical stress, all right? So all these different types of stresses, how does it affect the organism that lives in the ecosystem? Okay, next slide, please. So the first thing she did was, when we study the effect of ecotoxicology on animal, which is a broad theme, the next question is, what organism are we going to use? We can't possibly test on humans. So we're going to use an animal model, all right? Something that we can use in the laboratory, something that is reproducible in terms of results, something that is relevant with the environment. So when we talk about the use of model animal or the type of testing that we do, there are different types, there are different levels that we can test. We can test it at the molecular level. So this is using molecular biology techniques. We can use biochemical or even enzymatic pathways. So this is the effect of pollutant on enzymes, perhaps, all right? And then we can test it at cellular level. So we're sort of moving out from the micro stuff, looking at the cell. The effect of pollutants on cell structures, for example. Or we can look at the physiological, the histopathological, and behavioral effects. How does a pollutant affect the behavior of the organism? So that's another level that we can test. We can also use a whole organism, death, reproduction. So this would be referring to the toxic effect of pollutant on the organism, whether it kills the organism or not. And of course, the effect of toxic pollutant on reproduction of the animal. And lastly, we have population and community level as well as ecosystem. So the last 2 points sort of gray because that's big picture, right? That's something that we are still exploring. So we're looking just not one organism, but the entire spectrum of the ecosystem, right? So that's a big sort of broad definition on the effect of pollutant on the whole ecosystem. So basically, these are the different levels that we can test the effect of pollutants, all right? And then, of course, we have this other term called biomarkers. What are biomarkers? You know, this is basically a measurable stress response in an organism. So basically, when you're trying to test the effect of pollutant on an organism, the data that you generate needs to tell you whether the pollutant is toxic or not, and how do you measure it. So we bring in formulas, calculations as well. How do you translate behavior into a number, for example? So that's the mathematical part of it, all right? So all these sort of levels of measurement basically is to allow us to test the effect of pollutants on the organism using a specific biomarkers. Next slide, please. So this first part of the journey towards science education is basically on the development of a biomonitoring toolkit for assessing marine habitat health. So you might be asking me, why is she interested in ecotoxicology? The reason is because if we can use an animal model to test the health or to measure the health of an ecosystem, in this case, the marine environment. If you're not aware, Singapore is a small island surrounded by ocean. So then we are able to say, okay, maybe this area is not so healthy or healthy, based on the response of the pollutant on the organism. And that will give us an indicator, right, to tell us that maybe we should do something about this environment, or why, is this environment becoming toxic, for instance, all right? So the first part of this journey involves developing of a system, so to speak, a toolkit, to allow us to assess the health of the marine environment. And this spans about 3 years, from 2010 to 2013. Next slide, please. So here we are. We decided to, or rather she decided to pick on a species of mussels that is found everywhere in Singapore. And this has to do, this particular organism is the native green mussel, Perna viridis. So that's the animal model that we use to test the effect of pollutant on the organism. So basically the objective of this science part of it is to develop and apply biomarkers at various organismal levels to assess the marine habitat health and integrity, to establish a lab-based dose response assay using selected pollutants, establishing a monitoring program, and then eventually engage education groups as well as schools. So that's the last objective of it. But before we can do that, we need to make sure that the science is solid before we can even roll out to the school. All right? Next slide, please. So these are some of the biomarkers that we establish. All right? First is the clearance rate of the mussels. So if you are not aware, these mussels actually filter feed. For those of you with a biology background, these mussels actually filter feed in the ocean. So they pump in water and then they pump it out. In the process of it, they filter feed, all right? So clearance rate is how fast the organism filter the water through their body, all right? So you can measure the clearance rate of the organism itself. The second biomarker that we use is basal thread count. This has to do with, when the mussels get attached to a piece of rock, for example, they put up basal thread so that they can be attached to the rock. So the number of basal thread that the mussels put out is also a type of biomarkers. If the environment has lots of pollutant, the mussel doesn't put out too many threads, basal thread, as compared to a environment that is conducive for living. Then you find a lot of basal thread. So that's how we count. The next biomarkers will be fertilization assay. This is the effect of pollutant on the fertilization rate of the muscle. Neutral rate retention, NRRT, and she indicated as a gold star. It means that this assay is solid in terms of getting the result. There is no, there's no gray area. So this assay is actually a very good assay in terms of the effect of pollutant on the organism. And this particular assay of neutral rate retention, basically it measures the retention integrity of the cell that we extract from the muscle in retaining a rate dye. If the cell is healthy, it will retain the rate dye a lot longer than if a cell that is compromised, that is not healthy. All right, so that's the rate that we measure. And of course, there are other assays that we use. We have the ferric reducing antioxidant power, FREP assay. This is a test of the enzyme or the ability of the cell in detoxification. And then you have glycogen level, heat shock protein, total glutathione assay, etc. So these are different ways of measuring the stress response of the organism. It could be molecular all the way to cellular, as well as the whole organism in response to the pollutant. Next slide, please. So these are just preliminary results that she has established. This is a clearance rate result. This has been published already. So, I mean, it shows that it's not very clear. Basically, it shows, so the x-axis would be the concentration of the heavy metals. So this is done in the lab. So basically what we did was we have two tanks. We have one tank that is a control tank, meaning no pollutant. And then we have another tank that consists of the heavy metal. And of course, we can subject the organism to different concentration of the heavy metal. In this case, would be lead and copper, right? The effect of lead and copper, cadmium and copper on the organism, and is measured by the clearance rate. So even before we do this experiment, you can hypothesize already. You can already predict. In the control where there is can we just finish up and then do the translation? So a clearance rate basically is how fast the organism processes the water.
Yang sudah dari presentasi tadi dari Bapak Dr. Tan Lik Tong menjelaskan bahwa dalam sesi ini akan dibagi menjadi 3 bagian penjelasan. Yang pertama adalah establishing the science. Jadi bagian yang ini adalah memastikan bahwa bagian sainsnya itu sudah jelas, pasti dan benar. Kemudian yang kedua adalah translating into science education. Jadi menerjemahkan hasil riset tadi ke dalam pendidikan sains. Dan yang ketiga itu adalah citizen science atau masyarakat yang melek dengan sains. Dan kemudian setelah tadi penjelasan yang diberikan tadi tentang biologi, kemudian biomarkers dan lain-lain, itu adalah penjelasan tentang penelitian yang telah dilakukan oleh Dr. Tan Lik Tong. Dalam penelitiannya, karena beliau datang dari departemen yang berfokus pada sainsnya, karena tadi katanya dibagi 2 di NII bahwa ada yang sains, ada yang fokus di pendidikan, maka beliau menjelaskan aspek sainsnya di mana beliau berfokus dalam riset marine toxicology atau toksikologi ke lautan. Dan kemudian mungkin akan dilanjutkan lagi penjelasannya sehubungan dengan temuan-temuan yang ada. Jadi dikatakan juga tadi bahwa penelitiannya dilakukan dari tahun 2010 sampai 2013 untuk memastikan bahwa sains yang akan dibawa ke sekolah itu benar dan pasti, sehingga nanti siswa yang akan mendapatkan materinya itu benar-benar mendapatkan sains yang benar-benar digunakan di dunia STEM. Mungkin demikian. Maybe I think that's the summaries from what you have explained. Maybe you could continue. I think you could continue it in quite a length, and then I will try to summarize it. Okay.
Thank you very much. My apologies. I didn't realize I need to give her airtime. Okay. So anyway, so this slide basically is to show the correlation of the heavy metal effect on the clearance rate of the muscle. And without going into too much detail, you have to trust me on this. It does show some correlation. So that's the science behind it, right? Before we can even roll out to the school. Next slide, please. The next assay that we use is called the basal thread count, as I mentioned before, right? In a environment that the muscle, it's very happy in, they'll put out a lot of basal thread, right, to attach themselves on rocks, and then they start to filter feed. As compared to an environment where the muscle is in contact or exposure to pollutant, then the number of basal thread is limited or very few. They don't put out that many, all right? So that is, again, a very clear correlation that we can see between muscles that is subjected to heavy metal exposure as compared to muscles in a controlled environment, which is, no pollutants at all. Next slide, please. And then, as I mentioned earlier on, the neutral rate retention assay. Basically, this is a cell-based assay where we have to draw blood from the muscle, right? We have to draw blood using a syringe. We draw blood from the muscles, and then we extract the cell, and then we subject the cell to either pollutant or control, no pollutant. And we check whether the cell retain the dye or they release the dye in a very fast manner. So of course, in addition to subjecting the cell to the different pollutant, we also have to use a red dye so that you can test the effect of it. Next slide, please. And this is fertilization assay, where we test the effect of pollutant on fertilization. And again, it does shows a very nice correlation on the rate of, so you can see that the y-axis will be the different concentration of the pollutant, and the y-axis would be the percentage of unfertilized eggs. So you can see that with increasing concentration, the percentage of unfertilized eggs increases, all right? So which means that the muscles is highly stressed, and the fertilization rate is impacted by the high level of pollutant. Next slide, please. So those are just a few examples of bioassay that she developed, the science of it, to make sure that we get reproducible result before we can even introduce to the school. So why use biomarkers, you may ask? Well, because it's ease of employment, meaning that you can actually roll it out. Because one of the things that we have in mind is that when we introduce all these system or this bioassay to the school system, we have to bear in mind that it has to be relatively easy, quote unquote, right? It mustn't be too complicated, meaning that it shouldn't involve complex or sophisticated machines, so to speak. Because some of the schools, they don't have the machine. So we think about simple assay that the student can understand. cost-effective versus chemical analysis. So some of these biomarkers are relatively cheap, right? So if you want the school to continue running the program, you have to bear in mind the economy. It mustn't be too expensive. Rapid results. So again, you know, when you do the essay mustn't be too long, right? You have to factor in the time for the student to run the essay as well. So the essay shouldn't be, you know, it shouldn't be more than 1 week, so to speak. It should be at best maybe a few hours, you know, the longest maybe overnight. So you have to factor in time as well. First tier rapid assessment of habitat health. So this is where the relevance comes in. From the lab-based bioassay, how can you translate to the real-world situation now? Because the student might be asking, why, you know, what am I doing, or how is the experiments that I'm conducting, how does it relate to the real world in terms of pollutant effect on the ecosystem? Potential technique exportation to school and interest group, initially teachers only. So this, I will talk more about this later.
Oke. Mungkin bisa mundur slide-nya. Ya, mundur lagi. Oke, nah mulai dari slide yang ini tadi dijelaskan bahwa dengan menggunakan, jadi biomarkers itu adalah penanda organik dari yang diambil tadi dari kerang ya, dan kemudian yang hijau yang biasa kita makan. Nah itu kemudian diambil darahnya dan kemudian dilakukan uji menggunakan dilakukan uji untuk melihat apakah jika warna merahnya itu semakin terang, maka itu kemudian menunjukkan bahwa spesimennya itu dalam kondisi stres. Sedangkan pada saat dia sehat, maka dia akan berwarna pink. Next. Di sini juga merupakan salah satu contoh bagaimana untuk melihat keberadaan, is it the existence of pesticides, sir? Is it about the existence of pesticides?
The effect of pesticide on fertilization.
Oh,
Yeah.
okay.
So this one has to do, this has
Yes.
related to reproduction
Or related to the
of the,
reproduction
of the organism.
of the organism.
Yes.
Of the organism. Oh, okay, yes. And the higher the
So the
crop,
higher
does it
the
means
higher the concentration
of
of the
fertil-
pollutant,
of the pollutant
it affects the
Oh.
fertilization
based
rate.
on the fertilization assay. Oh, jadi dia melihat apakah terjadi, dia melihat tingginya ketercemaran suatu area tersebut berdasarkan tinggi rendahnya berdasarkan keberadaan fertilisasi tadi ya. Semakin banyak maka dia kondisi, berarti tempat itu, is it the more fertilizations means that there are more
So-
contamination?
So this is referring to the number of fertilized eggs.
Fertilized egg.
Yeah. So
The-
if the pollutant is high, then the rate of the number of fertilized
Fertilization?
eggs will be low
Or below, okay.
because it's impacted.
Because they're
The
died.
pollutant, yes. yes.
Jadi semakin banyak polutan maka semakin sedikit proses fertilisasi dan sebaliknya. Dan kemudian maybe next. Di sini juga dijelaskan pertanyaannya kan tadi kenapa harus susah-susah menggunakan biomarkers, karena biomarkers is mudah dipakai, it is of employment, dan dia juga cost efektif, murah, cepat, dan juga mudah dan berpotensi untuk diperkenalkan ke sekolah. It has a potential to export to school. Okay. Okay, I hope I'm not butchering a lot of them. Please continue.
All right, thank you. One of the things that I forgot to mention is that all this essay, we also involve undergraduate students. So this is where we start to get our students involved in these essays. These are undergraduate students in their 4th year, right? Because in their 4th year, they're supposed to do a final year project. And so in NIE, these students will be involved in some of these projects to get them involved, and then because like you, these students will eventually become teachers. So we train them also with a foresight, with an idea that hopefully, you know, these teachers will also roll out the program at the school. All right, so that's sustainable. I'm only one person, you know. To run this program, we need manpower. So we need to think long term in that sense. Next slide, please. So yeah, this is some of the output from the early studies, you know, some of the publications that we have, as well as a book on handbook of marine ecotoxicology techniques. So this handbook is basically a series of essays. You can download this for free. You don't have to buy it. It's freely available online. And this is important. Making, you know, publication available that is free. That will give the incentive, you know, for the teachers to use. If you have a cost factor to it, then, you know, there might be a little barrier to it. So yeah, so these are some of the publication that we have. One of the paper was published in the Asian Youth Journal of Biology, and this is something that I set up. I'm one of the chief editor of this journal, and this is to encourage students, high school student, university student, when they conduct a piece of research, we give them the option to publish in this journal as well. This is peer-reviewed journal, by the way. Next slide, please. Want to summarize before I go to part 2?
Yes. Jadi tadi dijelaskan bahwa yang dilakukan penelitian dilakukan tadi itu adalah hasil dari riset mahasiswa S1, is from bachelor students ya. Dan itu kemudian dipublikasikan dalam berbagai macam bentuk, ada yang dalam bentuk jurnal dan ada juga dalam bentuk handbook ataupun buku. Yang dimaksud handbook itu adalah buku manual ya tentang teknik-teknik dalam ekotoksikologi di kelautan. Jadi yang ingin digarisbawahi adalah ini merupakan hasil riset yang kemudian dipublikasikan. Saya kira itu sesuatu yang juga dilakukan oleh mahasiswa-mahasiswa kita pada saat kita meluluskan mereka. Dan selanjutnya yang akan dibahas itu adalah tentang bagaimana mentranslate semua penelitian tersebut ke dalam pendidikan. Please continue, sir.
So I just finished with part 1, which is establishing the science. Now let me move on to part 2, which is the transition or the translational part of it into science education. And this stems from, this is where I come in, right? And this is part of a grant that I received from MOE, the Ministry of Education in Singapore. And the title of this project is called Engaging Secondary School Students in Authentic Research Project based on Environmental Science Theme. So that's the title of the project. And it spans about 3 years, this particular project. And I'm the co-PI, and then again, Dr. Beverly Goh and Subramaniam, who were my co-PI. Next slide, please. So yes, the use of project-based learning to enhance understanding of the nature of science and scientific inquiry. So this is something that one of the objectives is the use of PBL, project-based learning, to enhance students, high school students, understanding of NOS as well as scientific inquiry. All right, so that's one of the objectives. And of course, part of that objective, we're also thinking of involving science teachers, not just a student. So this is like a partnership with a school where we involve science teachers. So this school teachers engage as partners in teacher-scientist partnership model. This is a model that we came up with. Usually it's engaging the student directly, but we thought to involve the teachers as well. So it's a tripartite arrangement where we have the teacher coming in, and then we also involve the students eventually. I'll talk more about the program later. Development of effective and sustainable PBL program within the Singapore school system. So hopefully that this model that we came up with will be sustainable in that sense. Next slide, please. So this is just examples of the program. The program, I should say the workshop. The workshop is over 3 days. On the first day, we only invite the teachers. So the teachers will come to NIE in the laboratory and they learn the various essay that was already established in the science part of it. So we teach the teachers how to run the essay. Why do we do that? Because we want them to understand the basis of the essay, not just the student. The reason is so that they can help us, right, to guide the student too. Again, we try to skill up, right, in terms of manpower, in terms of capacity, not just for the student, but also as a professional development, you can see it that way, for the teachers, all right? So these are some of the pictures that involve the teachers, and then the day 2 and day 3, that's where we start to involve their students, all right, from different schools, not just one school. So we involve the teachers on the first day, and then subsequent second and third day, we involve the students. Next slide, please. So one of the things that we train, not just the student, but also the teachers, would be providing skill training on instrumentation. So this is related to STEM techniques. Part of STEM education has to do with training students and teachers on how to use the instrument. A lot of time we assume that they know how to use micropipette. In fact, some of the teachers don't even know how to properly use a micropipette. So we cannot assume that they know, because sometimes they don't use that kind of instrument in the school. So one of the first things that we do is to make sure they know the basics, right? We cannot assume that they know. So we cover things like the use of compound microscope. You might think that it's such a simple thing, they should know, but not really. Some of the students have never used it, so we need to train them as well. The use of micropipette, and also instrument or technique that is relevant to the assay, to the bioassay. Because some of these techniques are specialized that the students need to learn.
Thank you. Mundur. Satu slide lagi. Oke. Ya, mungkin sebelum kita masuk yang ke sini ya, izin men-summarize dulu tentang riset yang telah dilakukan. Jadi risetnya itu kan tentang toksikologi di area laut ya. Yang dicek itu adalah pengujian kadar logam. So assay itu adalah pengujian kadar logam, dan itu dilakukan dengan menggunakan darah dari mussels tadi, dan juga melihat tingkat fertilisasinya. Nah, itu sudah dibuat penelitiannya, dan kemudian dibuatlah project yang selanjutnya, yaitu engaging secondary school students ini. Jadi ini tentang project dari pihak dari Dr. Tan Lik Tong untuk bersama guru melakukan riset otentik bersama siswa. Jadi guru, siswa, dan peneliti untuk kemudian bisa dikatakan mereplikasi apa yang sudah dilakukan tadi oleh para saintis. Next. Nah, di sini mungkin tujuannya itu kenapa jadi harus seperti itu, karena yang kita ingin mencoba memperkenalkan tentang konsep nature of science, atau tentang bagaimana sih science itu dilaksanakan atau dilakukan, dan juga tentang scientific inquiry. Karena selama ini kalau misalkan di ruang kelas pasti bahasannya itu tentang penelitian itu pasti yang sederhana gitu ya. Meanwhile yang ini adalah proses riset yang sebenarnya, dan siswa itu diajak untuk bersama-sama peneliti melakukan hal yang sama yang sudah dilakukan oleh peneliti. Dan dengan adanya proyek ini mereka juga mencoba untuk mengembangkan suatu usaha yang sustainable dan yang berkelanjutan dan efektif dalam hal pelaksanaan project based learning antara sekolah dan juga universitas. Next. Nah, di sini adalah dokumentasi workshop yang dilakukan di sekolah. Jadi siswanya itu diajak untuk mengulang lah istilahnya melalui workshop tentang penelitian yang telah dilakukan oleh para peneliti, dan ini yang diajak adalah siswa SMP dan juga SMA, dan workshop ini dilaksanakan dari tahun 2014 sampai 2016, melibatkan 23 sekolah dan lebih dari 40 guru dan hingga lebih dari 160 siswa. Next. Dalam workshop ini mereka jadi benar-benar diajak menjadi seorang saintis. Kenapa? Karena itu menggunakan berbagai macam peralatan yang digunakan oleh ilmuwan, yaitu menggunakan mikroskop seperti yang dilihat di layar dan juga menggunakan mikropipet, menggunakan berbagai macam teknologi yang biasanya hanya yang tidak digunakan di sekolah. Dan kesempatan inilah yang mungkin terjadi ketika ada kerjasama antara guru, siswa, dan juga peneliti. Izin mungkin kalau mahasiswa di Prodi Pendidikan IPA pernah belajar mata kuliah Pendidikan IPA Terintegrasi, ini adalah model ke-10 dari modelnya Robin Fogarty. Mungkin itu, I think that's.
Thank you. Next slide, please. So yeah, so this is where we have eventually, when we get the student to do the project, the STEM activity within 3 days, we also get them to present their result as a poster presentation, right? So these are just some pictures of students standing by their poster. At a poster presentation, we organize it as a mini symposium for the students. So they come to NIE again, and then we have judges go around, asking questions. The student will first of all present the result of their poster, and then we ask questions, and based on that we can evaluate in terms of their understanding of what they do. All right, next slide, please. So again, these are some of the publications that were published in the Asian Youth Journal of Biology. Next slide, please. Okay. So, shall I continue? Okay. So the part 3, proof of concept citizen science. And this is the title of this part 3 is development of a multi-organism and multi-level biomarker system for biomonitoring of health, of marine ecosystem health. This, in the beginning, we just focus on mussels. And this third part here is the two things to this third part. First is, can we now involve other types of organisms, not just mussels? Right? If we think about ecosystem, it consists of different types. The second thing is that, can we also involve citizen science, students as citizen science, and relating that to the actual environment? So again, this part took about 2 years. Next slide, please. So basically, the two main objectives of this proof of concept in citizen science is to explore the use of biomarkers in other organisms, not just mussels, at all trophic levels of Singapore natural marine food chain. And then to expand the application of biomonitoring to schools in Singapore. Hopefully, this is something still in development, upper primary to tertiary level, and integration as a comprehensive citizen science program involving relevant government agencies. Next slide, please. So this is a range of organisms that we start to sort of explore into. Not just mussels, we also use the Oncidium, which is a kind of mollusk. We also use starfish. We also use sea cucumber. All right. Next slide, please. So back to the science again, back to square one again. Every time we start with a new organism, we need to validate, right, whether there's any correlation of that organism in response to the pollutant or not, right? So we need to establish the science of that. So these are some of the results that we obtain. Next slide, please. And this one of the behavior. Remember the biomarkers that we talked about? There's one about testing the pollutant on behavior, So this is one example here. So this is the writing behavior of a starfish. So what we do is a very simple experiment. We basically flip the starfish upside down. So once you do that, after a while, the starfish will write itself. But if the starfish is stressed, the time for the starfish to turn it over is longer as compared to a healthy starfish. All right? Next slide, please. So,
mundur lagi ya. Oke, thank you. Ini mungkin adalah contoh ketika siswa pada saat mereka sudah melaksanakan workshop tersebut, mereka membuat poster dan kemudian mempresentasikan apa yang sudah mereka lakukan, dan itu ada dari beberapa sekolah ya. Dan kemudian next, ini juga adalah contoh artikel yang dipublikasikan sehubungan dengan pelaksanaan proyek tadi. Cuma mungkin memang belum oke. Next. Baru kemudian masuk ke bagian ketiga dari presentasi ini tentang proof of concept dari citizen science. Maybe I will I'm not sure yet, but I will proof of concept of citizen science. Does it mean like this is the more people involving in this project?
Yeah, basically it's a skill up of the project involving not just more students, but also government agency as well as other types of organism.
Okay, and you're going to explain that as a conclusive part of this presentation, or?
Yeah, of course.
Okay. Then I will just skip that.
Okay.
I will know. Yes.
Next slide, please. Okay. Yeah. Thank you. So basically, this is where we start to engage government agency, right, to come in. So this is the National Parks, NParks. This is a government agency where they are involved in the environment. Right, from botanic gardens to the ocean to the wayside trees, etc. So they are involved in that aspect of it. Now the reason why we involve them, a very simple reason, because some of these sites are restricted. We need to get permission from NParks before we allow the students to get to this site. And the other reason is also, which became a realization to us, is that some of the workers at the government agency are also interested in our biomarkers. So we also involve these officials from NParks, and we also train them, right, in the bioassays that we established. So this is not just training of students, but also teachers as well as officials from the national parks. And also deployment of muscles to NParks field sites. So this is where the relevance comes in. All along we've been conducting the research in the lab setting. But we want to introduce the authentic nature of that, is that can we now bring the mussels or other types of organism into the field and then bring it back again and then run assays on them. So by doing that, you're actually trying to teach the student, the experiment methods, steps to experiment methods in terms of how do you come up with, the sequence of experiments, or what are the process of conducting experiment. So the students will need to think of a research program involving a particular organism. So this is where there's a lot of interaction with the student, trying to discuss. The student will have some ideas, but sometimes the ideas is way out there, so we need to rein it back. We need to bring it back, and we need to bring in the science as well. So this is where we begin to teach the student how to formulate an experiment. Because the students have no idea what an experiment is, especially one that involves the natural environment. So we need to teach the student as well. So this is a period where there's a lot of engagement with the student, with the high school student, that's what I mean. And of course, our own student, the undergraduate student, will have some experience of it already, and they will help as well, right? Because in their final year project, they have gone through that process, so they know what is expected. So we also get them involved in the teaching of the scientific inquiry method to the high school student. Guided walks at deployment location focus on nature appreciation. So these are some of the benefits, you know, when we try to bring our high school student to the field, is that most of the time, you know, they're always on their handphone, you know, they're always at home. They have no idea what nature is. So this is a time where we also introduce to them appreciation of nature. That, you know, there are other things out there that you can explore, other ecosystem to explore, etc. So this is where we bring in the love of nature, biophilia, all right, to the student. That we need to take care of our environment. Otherwise, you know, it will lead to some detrimental effect if we don't take care of the environment. So we also introduce the concept of, you know, caretaker, for example, of the environment. Muscles collected and biomarkers analyzed back in the lab. So this is part of the experiment setup where the student will deploy the organism at specific sites around Singapore, and then we have to bring it back into the lab, and then we test it again. So this spans not 1 day, not 2 days, but it could be several weeks. So we really need to factor in time for the student. Bear in mind that this is extra for the student. This is not part of their curriculum. So we need to factor in, you know, when is their holidays, etc. So it's massive planning, and of course the teachers will help as well in planning. And lastly, data provided to MParks. So there's a lot of utility for this. This is where we begin to go into citizen science. Is that a lot of this data generated by the student can also be used by MParks. So this is where, you know, we need to make sure that the numbers, the data is legit. You know, it's not just, you know, generated any old how, but the data that's generated by the student will be useful for MParks.
Thank you. Oh, mundur agak jauh kembali ke yang tadi, Pak. Iya. Oke, jadi yang dijelaskan tadi ini adalah tentang bagian dari setelah sainsnya diaplikasikan oleh siswa di kelas melalui workshop karena tadi pada saat yang pertama kan itu adalah pengembangan tentang biomarker. Yang kedua adalah setelah mengetahui bahwa kita bisa melihat dari kerang apakah area tersebut banyak mengandung logam, kemudian juga bisa melihat dari fertilisasinya tadi, maka siswanya diajari dengan cara melihat komponen itulah kita bisa mengetahui bahwa adanya ketercemaran di suatu area. Nah, jadi berarti kan di situ ada yang awalnya hanya sejumlah sedikit saintis jadi banyak sekolah karena tadi involving a lot of schools. Nah, selanjutnya adalah citizen science-nya, ini adalah masyarakat yang melek dengan sains. Di sini mereka berusaha untuk mengeksplorasi kegunaan biomarker tadi untuk kemudian melakukan monitoring terhadap lingkungan tertentu. Dan itu melibatkan sekolah. Jadi peneliti melibatkan sekolah dan siswa-siswanya itu melakukan biomonitoring di tempat yang dipilih. Next. Ini adalah spesies-spesies yang lain yang memungkinkan untuk dijadikan sebagai biomarkers. Selain dari kerang tadi juga ada teripang, kemudian juga ada bintang laut dan lain-lain. Next. Nah, di sini menunjukkan hasil penelitian selanjutnya bahwa tadi dikatakan kerang bisa nih jadi biomarker, lalu kemudian spesies yang lain bagaimana kita melihatnya. Ternyata pada bintang laut itu jari-jarinya menjadi lebih panjang gitu ketika suatu area tersebut lebih terkontaminasi dengan logam. Dan hasil-hasil lainnya yang kemudian bisa menjadi pegangan bagi peneliti, siswa untuk mengatakan bahwa oke, biomarker ini bisa kita pakai loh untuk mendeteksi suatu daerah itu tercemar atau tidak. Next. Ini contohnya ya tadi yang dijelaskan bahwa dia bisa, kalau misalkan normal dia pendek, tapi kalau misalkan dia dalam kondisi tercemar nanti itu akan menjadi lebih panjang anunya, bagian potongan tubuhnya. Next. Bagian yang ini menjelaskan tentang apa sih kemudian yang dilakukan oleh saintis, guru, sekolah, dan melibatkan taman nasional atau national parks yang ada di Singapura. Jadi mereka ada 5 kegiatan yang dilakukan. Yang pertama itu kan tadi melatih guru dan siswanya, kemudian setelah mereka dilatih dikumpulkanlah spesies-spesies tadi. Yang ketiga dikumpulkan spesies tadi dan spesies tersebut disebarkan di area taman nasional. Yang ketiga adalah kegiatan jalan-jalan lah di lingkungan tempat dilepasnya spesies tersebut untuk kemudian menciptakan rasa cinta terhadap lingkungan hidup. Yang keempat, setelah beberapa bulan, mungkin 2-4 bulan, spesies yang sudah dilepas tadi itu diambil lagi untuk kemudian dilakukan kajian untuk melihat apakah terjadi apakah terdapat kontaminasi di area yang menjadi tempat mereka melakukan penelitian atau monitoring. Dan yang terakhir adalah setelah diambil kemudian datanya diolah dan dikumpulkan, semua data yang dikumpulkan tadi itu diberikan kembali kepada Taman Nasional. Jadi menunjukkan bahwa ada kolaborasi antara banyak pihak dan tidak hanya sekolah saja, tidak hanya ilmuwan saja, tapi kita memiliki masyarakat ilmiah atau citizen science.
Next slide, please. So this is just a map of Singapore with all the study sites, around Singapore. A few on the north, and then just one or two in the south of Singapore. And so this program, over 2-year period, involved about 44 students, 118 pupils from 21 secondary schools. Next slide, please. So again, this is just some pictures of the students coming into lab. So after they deploy the mussels or other types of organism, they bring it back to the lab and then they carry out testing. Next slide, please. Yeah, and so this is some of the some pictures that shows the study site, where on the left side, leftmost corner, you have the deployment of mussels at the study site, and then we leave it there for maybe a week or two, and then together with the teachers and the students, they'll go and retrieve the organism back to the lab, and then they conduct the experiments. Next slide, please. So yeah, so this is the same. Next slide, please. So what was data collected by our young citizen scientists rigorous? So again, in order for the data to be useful for NParks, we have to make sure that the data is solid, is kosher. The short of it is yes, especially if you use neutral rate retention time assay, the NRRT assay. That's why I put a star there, the gold star, the gold standard. So that's the assay that really gives robust result, when you carry out the assay. Next slide, please. And of course, the student also get to present their work to the President of Singapore, Tony Tan, back in 2015 at a Sisters Island Marine Park Public Gallery more than 5 years ago. So this is where the student will also present the results of their work to the ministers as well as president. Next slide, please. Okay, maybe I stop here. Would you like to translate?
Sorry, I was reading. Thank you. Jadi ini sebenarnya tampilan dari proses kegiatannya ini adalah pada saat melepaskan kerang. Jadi itu ada, mungkin bisa dilihat dan dibayangkan ya apa yang dilakukan di sana supaya kerang itu bisa diambil lagi setelah sekian bulan dan gak hilang. Next, thank you. Ini juga adalah proses ketika siswanya melakukan pengambilan data sesuai dengan apa yang sudah dilakukan oleh peneliti sebelumnya. Next. Next please. Ini adalah data yang pertanyaannya adalah apakah kemudian data yang diambil oleh siswanya ini rigorous atau datanya itu bagus gitu. Dan ini menunjuk di sini bisa terlihat bahwa ada 3 tempat yang yang diambil datanya. Itu ada yang area kontrol, ada daerah Punggul dan juga Sungai Buloh. Dan itu menunjukkan ada perbedaan hasil karena memang berbeda tempat, berbeda kondisi pencemarannya. Next. Nah di sini impactnya mungkin yang terlihat namanya kita mengukur impact ya, kita melihat dari angka dan datanya itu menunjukkan bahwa ada 178 masyarakat ilmuwan yang dari masyarakat yang berpartisipasi dalam upaya biomonitoring ini. Ada banyak pihak yang terlibat dan juga di layar bisa dilihat salah satu siswa itu mendemonstrasikan proses teknik yang digunakan dalam program ini kepada presiden. Jadi proyek ini diapresiasi di Singapura sebagai salah satu proyek yang bisa tidak hanya menghasilkan ilmuwan saja tapi menghasilkan masyarakat ilmiah. Seperti itu. Next. Please. Okay. Thank you.
So these are some of the learning points from that long period of starting from establishing the science involving teachers and students. So these are some of the, as we reflect as educator, what are some of the points that are important for us. One of the things that we talk about or think about is when we consider a coastal management from a different perspective, is that harnessing citizen scientists to aid in biomonitoring as well as data collection. So this is where we start to sort of think about, can we involve the general public to generate, to help us monitor the environment, not just the scientists, all right? And from that perspective, then you start to think about how we're going to train them, all right? Because they need training first before they can even, help you with biomonitoring and to take on the role as citizen scientists. Is that how do you train them properly, all right? Importance of collaboration between scientists, government agency, and school to ensure rigorous method and analysis. This is where, the interaction of not from the student perspective, the interaction with the scientist is good, is an important one, of course, but then the interaction with the government agency adds the relevance to it, in terms of why are they doing this to begin with? How does it constitute, or how would their work, be useful in a national sense, all right? So that would take pride, for the student to take pride in their work and take ownership of their project, all right? So this is something important for from the student perspective. And thirdly, the sound biomonitoring requires an array of just about half a dozen biomarkers assays utilizing local species. Easy. So, I mean, this easy is a relative term, all right? And before we even embark on this project, we have to make sure that the science is sound. And that takes time. It could be a few years, to develop it. So, next slide, please. So the take-home message, or the take-home lessons in the journey of applying science to citizen science, involving teachers and students, I leave you with 4 points. Lose the jargon and speak the lingo, pitching it right. So as a university researcher, sometimes when we talk to high school student, we assume that they know certain terms. I remember a week ago before coming here, I was engaging some high school student from a relatively good school, and I use the word fluorescence, then they ask me, what is fluorescence? So I assume they understood what fluorescence is because they're coming from a good school, but you cannot assume that, So you need to be very careful, especially when you have, at the university level, when you pitch your idea or when you talk about certain things to the student, the high school student, you need to be very aware of the term that you use. You need to simplify things a lot. Low limit to learning or student's ability. Most of the time, we second guess the student. We think that the student cannot do this. When we develop the essay, we already, in our mind, we're like, oh, student cannot do this. It's going to be very difficult for a student, not necessary. When one of the essay involved drawing of muscles blood using syringe, we thought, okay, no problem for us, but for students would be difficult. But lo and behold, when we taught this to the student, some of them were able to draw blood effortlessly. So don't put boundary to what the students can do. They can do a lot of amazing things, all right? Have faith in them. Of course, you have the occasional student who will, instead of drawing blood, they pierce their fingers or whatnot. So make sure they sign an indemnity form. Train the trainers, teachers as confident science mentors. So the teachers will also participate in this program and take ownership as well. And also as part of their development, professional development. Because most of the time teachers, they do teaching in the school, they may not have the time to go back to school themselves. So this is a perfect opportunity to train the teachers in order to upgrade their skills, so to speak. And this is something that we do continuously, not just in my lab, but also at NIE, the National Institute of Education, where we have an array of programs for teachers, not just teachers, but also school principals in terms of leadership roles, etc. Revise, and share freely. This is something I mentioned already, that when you develop any tools or any manual, make it available for free. If you put a cost to it, then it will hinder the school, especially those who cannot afford it. So make sure that your material, your manual, can be available freely for other educators. And also, you guys can actually download this manual and run it in your lab.
Okay. Mundur, Pak. Thank you. Di slide ini itu ada tulisan bahwa consider coastal management from a different perspective. Jadi kita melihat tentang manajemen pesisir itu dari perspektif yang berbeda. Bagaimana dia berbeda? Karena proyek ini dilakukan tidak hanya sendiri saja, tapi juga melibatkan banyak pihak. Jadi dia menggunakan kekuatan masyarakat ilmiah untuk membantu proses biomonitoring dan pengumpulan data. Yang kedua adalah pentingnya kolaborasi. Nah, kolaborasi ini yang dilakukan itu melibatkan sekolah, ilmuwan, dan juga lembaga pemerintahan untuk memastikan bahwa datanya itu bagus atau rigorous. Kenapa? Karena semua orang take ownership atau merasa memiliki atas keberadaan program ini. Karena ini bukan hanya, ini hanya tugas aja nih misalnya, tapi tujuannya adalah ini nanti datanya setelah dikumpulkan akan dipakai oleh Taman Nasional, dipakai oleh negara, sehingga penting untuk datanya itu, maka kualitas datanya itu terjaga karena adanya proses kolaborasi yang melibatkan banyak pihak. Bagian yang ketiga itu dikatakan yang namanya mudah itu kan relatif gitu, tapi kemudian karena ada proses pelatihan dan lain sebagainya, maka harusnya proses ini akan lebih mudah untuk dilaksanakan. Next please. Nah, take home lesson-nya itu ada 4 yang tentang bagaimana dalam perjalanan untuk mengaplikasikan sains kepada masyarakat. Yang pertama adalah ketika kita mencoba untuk memperkenalkan hasil penelitian ini, maka mungkin kita harus meninggalkan jargon atau meninggalkan istilah-istilah ilmiah yang mungkin bagi orang awam itu membuat sulit dan membuat mereka terdemotivasi untuk berpartisipasi dalam proyek ini. Sehingga kalau ingin semua orang berpartisipasi, jadi harus menggunakan bahasa yang sama. Jadi mungkin kita perlu menyesuaikan perspektif sesuai dengan pihak yang akan kita ajak berkolaborasi. Dan yang kedua adalah tidak ada batasan dalam proses belajarnya siswa, karena tadi siswa yang awalnya belum pernah penelitian, tidak tahu apa itu ya eksperimen, akhirnya mereka bisa melakukan eksperimen bersama dengan tuntunan dari guru-guru dan juga peneliti yang aktif di dalam kegiatan ini. Dan yang juga pentingnya itu adalah ketiga, yaitu melatih gurunya, melatih trainersnya, dan membangun kepercayaan diri agar gurunya itu merasa aku bisa mengajarkan ini nih ke siswaku, sehingga yang proses osmosis pengetahuan dari saintis guru ke siswanya itu terjadi dengan lancar. Yang keempat, apapun yang sudah dilakukan itu terus dilakukan perbaikan dan juga dibagikan secara gratis agar semua orang bisa mengakses itu dan belajar bersama-sama dan bisa melibatkan lebih banyak pihak lagi. Next, please, or is it the end? Next, please.
Next slide, please. I think it might be the last slide. Next slide, please. Yeah. So, yeah, speaking the lingo, pitching it right, what do we mean by that? You know, when we talk about the science part of it, biomarkers, these are all big terms that the students have no idea. Ecotoxicology, right? These are all big terms that the student might not understand. So when we introduce a topic to the student, we need to pitch it right, meaning that perhaps you have to use analogy of, you know, doctors monitoring our health. How do they do that, right? So we as the organism, now the doctor is trying to monitor our health. What are some of the signs or some of the measurements that it takes? It could be temperature, it could be blood pressure, etc. So we start from there first to explain to the student. This is a high school student, 14 years old, 15 years old. They have no idea what ecotoxicology is. So we need to start with something basic that they can understand, because most, if not all of them, have seen a doctor and they know what is involved in the process of that. So yeah, so we talk about this, and then we slowly start to bring in, you know, the analogy of that, and then we bring in ecotoxicology so that they can understand the significance of it. What to expect from this workshop, and this is where the marketing comes in, right? To have the buy-in from the school, all right? Is that, you know, you will learn cool techniques and conduct real STEM research. This is something that we advertise to the school as well, you know? So something that the school can benefit. How does it benefit the teachers? How does it value add to the school, so to speak? So it's that you will learn cool techniques and conduct real STEM research and interaction with scientists. You may present your research at competition and scientific gathering. So these are some of the enticement for the teachers as well as the student, for them to participate in STEM education. You may talk about your research and impress someone of importance, for example, the Singapore president, which actually happened. Next slide, please. So low limits in learning or students' ability. This is something that I mentioned before, is that, you know, this is the left picture shows, that's Dr. Beverly Goh. She's demonstrating to the students on how to draw blood from the muscles. So this is something that we thought the student cannot do, but actually they can do it very well. So, so yeah, even secondary school pupils were able to master the technique in a very short period of time. Next slide, please. Train the teachers as confident science mentor. This is, again, trying to provide useful resources for the teachers. So in addition to training them, the essay, we also provide some of the resources for free. For example, of course, the book is free. The use of an Excel spreadsheet with statistics, so they can actually perform statistics based on the data that they generate, right? So this statistic package is free for the teachers too, and it's easy to use. This was some. Okay, so this is referring to when students come to us, they expect the result to be 0 and 1. You know, so they have no, how can we still get result, you know, in terms of thinking of 0 and 1, when we're dealing with the environment, you know, there are other things to consider. It's not so straightforward. Sometimes the data is not straightforward, and that's where critical thinking comes in, right? You need to teach them, you need to tell them that, you know, if your data is anomaly, right, it doesn't fit the hypothesis that you came up with, why? You know, so this is where the student will start to think, will start to measure other parameters, etc. All right, so this is where, you know, letting the student sort of accept different shades of gray. Nothing is black and white. Sometimes the result can be gray. If it's gray, what does that mean? All right, next slide, please. So yeah, revise, and share. The books have gone through many versions. So in the beginning, you know, it was meant for scientists and, you know, higher degree educator. And then as we revise, we took okay, thank you. So next. So that's it. Thank you.
Mundur ya, cek, oke. Lagi 2 slide ya, oke. Ini mungkin tadi sudah di list ya tentang apa saja yang harus dilakukan. Yang pertama itu speaking the lingo, pitching it right, artinya bagaimana cara menyampaikannya dengan tepat. Nah, di sini contoh yang diberikan itu seperti ini. Bagaimana seorang dokter itu bisa memonitor kesehatan kita, maka pasti ada yang diambil, pasti ada yang diukur. Dan kemudian pasti ada sampel yang diambil, yaitu seperti darah, urin, dan juga jaringan-jaringan yang ada pada tubuh. Nah, kemudian itu apa yang dilakukan? Misalnya pada darah kita bisa bisa melihat enzimnya sebagai biomarker. Kemudian kita juga melihat ada biomarker kanker, dan yang ketiga juga kita juga bisa melihat apakah terjadi infeksi bakteria. Jadi mirip seperti ketika dokter memonitor manusia, maka kita juga bisa melakukan itu kepada kerang dan spesies-spesies lain. Nah, begitu cara maksudnya cara menyampaikannya kepada siswa, nggak kemudian langsung biomarker adalah ini gitu, tapi dicoba cari analogi yang bisa membuat siswanya itu bisa memahami apa yang dilakukan oleh para ilmuwan. Nah, selanjutnya juga dijelaskan kepada siswanya supaya mereka termotivasi. Yang pertama tadi disampaikan bahwa kalau kalian ikut workshop ini, maka kalian bisa mempelajari teknik STEM yang keren. Kemudian kamu bisa mempresentasikan riset kamu dalam kompetisi atau bisa membuat poster dan tetap bisa mengikuti banyak pertemuan ilmiah. Dan yang ketiga, mungkin kamu bisa memukau seseorang yang penting. Mungkin bahkan sampai bisa ngobrol dengan presiden kalau kamu melakukan penelitian ini. Jadi caranya itu menyampaikan kepada siswanya, kepada gurunya itu harus dimoderasi. Next please. Yes. Nah di sini bisa dilihat bahwa anak SMA ya. Oh ini SMP. Anak SMP pun bisa melakukan penelitian ini. Next please. Di sini juga ditunjukkan bahwa guru-guru yang membantu proses pengambilan data atau biomonitoring ini telah dibantu dengan dikasih file templates dan juga berbagai macam bantuan yang bisa mempermudah guru untuk memproses data yang sudah dikumpulkan oleh siswa. Dari yang terakhir, next please, adalah menjelaskan kepada siswa bahwa segala sesuatu itu sifatnya relatif. Dia nggak akan jelas salah benar hitam atau putih, tapi kemudian data itu dikumpulkan baru kemudian sama-sama dianalisis. Next please. Dan yang ini adalah revisi terus menerus dan juga dibagikan. Di sini ada 3 versi dari buku yang sudah dibuat tadi kan pada tahun 2013 ya. I think the first book is in 2013 ya. Dari tahun 2013 sampai tahun 2016 itu sudah ada 3 versi dari buku yang berhubungan dengan bagaimana cara melakukan deteksi pencemaran logam tadi. Next please. Demikian adalah akhir dari presentasi dari Dr. Tan Lik Tong. Please once more give round of applause for Dr. Tan Lik Tong. We're going to the question and answer session. Okay. I will be taking 2 questions first. Saya akan mengambil 2 pertanyaan. You can ask in Indonesia or English. Please mention your name, sebutkan nama, prodi apa, dan berikan pertanyaan yang singkat saja. Yes, please.
Thank you very much for your course for today. I'm very appreciated your course. Maybe can inspire our research, the next research here. The one is establish science. The second is implement to science education, and the third is make citizen science. Yeah. So apa ya bahasa Indonesianya ya? Ever implement STEM PGBL in our microbiology learning. The science is cultivate microba. We saw colony of microba, and the second science we learn about coliform. Yeah. Lalu the technology we use micropipette, we use autoclave, and we confuse the engineering is apa gitu ya. Engineeringnya itu seperti apa gitu. Jadi ketika kami sebagai guru atau sebagai dosen, I'm sorry, I'm not good enough to speak English. So why we, our teacher, we as a teacher implement STEM in learning? Apakah itu apa ya bahasa Indonesianya ya? Apakah kita itu menerjemahkan STEM itu per each part STEM we describe in learning? Gimana ya? Apakah tiap pembelajaran STEM itu harus memenuhi kaidah dari sains, teknologi, engineering, and matematik? Seperti itu. Lalu saya ingin tanya bagaimana penjelasan secara singkat mengenai neutral rate retention dalam kerang itu. Bagaimana cara simple, the tool, the material, and the procedure in simple way about neutral rate retention. Thank you very much.
Thank you for the questions. Let me maybe a little bit summary of the question from Ms. Lutfi. So she was asking whether we are supposed to separate science, technology, engineering, and mathematics, because she was confused when she was implementing STEM. What is the engineering part of
sometimes when you implement STEM education, right, we're so fixated with, the need to implement all these different components together, and sort of losing sight of from the student perspective, right? And sometimes you might, sort of downplay certain element of the STEM procedure, because ultimately, what are you trying to achieve for the student? The STEM, the science of it, the mathematics or the engineering, or do you have to encompass everything? So it can be very confusing. So you need to start baby step, small steps. The engineering part of it has to do with design, right? So if the project, if the activity doesn't really involve design, or maybe it's not as important, right, then it's okay. Ultimately, what are you trying to achieve for the student? To get them be confident as in terms of running experiment, to think about the data in a critical manner, etc. That's good. You don't have to incorporate every single thing and force it into an activity that sometimes becomes very artificial, and then it's like the teaching part is you get lost in translation, so to speak. So don't be so fixated with, okay, I need to implement engineering as well, otherwise it's not STEM education. Not necessarily. The way I see it, STEM education, you ask 10 people what's the definition of STEM education, you have 10 different answers, right? So what does STEM means to you, all right? And then from there, harnessing on your own strength, and then come up with something that is authentic, that is useful and relevant for the student. So don't be so stressed about having to implement everything. Some of the activity doesn't really involve, or very minimal in terms of engineering part of it. The engineering could be, example of it could be, again, I mentioned design, the structure itself, or it could be something computational. It could be, an app. It could be coding. Right? So that's also part of engineering, so to speak. So it depends on how you define, the STEM education in your own sort of capacity, because sometimes you need also to involve other discipline, other people of different background. So your background is in biology, so you might involve, colleagues or your friends in mathematics, and then try to sort of come up with something, bounce off ideas with each other. What is the mathematical part of it, etc. What can be helpful is that if you're a beginner practitioner, is to look at model packages. Go online, STEM packages. Look at what is in those packages. Have some idea first, instead of starting cold. Sometimes starting from scratch without any idea of what's out there is difficult. So you need to educate yourself as well, what's out there, and see whether you can make use of some of the existing resources. You don't have to create something all the time. Make use of existing resources and then tweak it to your own context to make it relevant, not just for yourself, but also to your students. Because ultimately, they are the focus, right? And of course, what are your learning outcomes from that STEM activity? What do you hope to achieve for your student? Is it competency? Is it critical thinking? Or both? Is it creativity? What do you mean by creativity, right? I was talking to my science educators in my department. Creativity, there are 4 or 5 types of creativity. To me, I thought there's only one. So they can come up with 4 different types of creativity, and they'll try to explain to me the definition of creativity. And after explaining, I'm like, they all sound the same to me. So sometimes you can get, even among peers, we confuse each other, right? PBL, inquiry-based learning. Maybe there's subtle differences, but to a person with no background, they all sound the same. So my background, my research is not in science education per se. My background is as a chemist. So I approach it from a practitioner perspective, So we're all coming from different background, different sort of perspective. and we create a rich learning environment for the student. There's no one way, there's no one correct way. There are multiple integrated ways of learning, you know. So that's, I guess, my advice to you. You know, don't be too stressed. Try to incorporate everything into STEM education. Start with what's available first, and try to make it your own. Sometimes the method can be obtained from what has been published already. You know, so in this particular example, the program itself, what is important is the choice of the organism, the model organism. So I remember this talk was presented not by me, by Dr. Goh at another conference. And one of the questions was, how do we start biomonitoring program in my own country, right? So the first thing to do is what's in your country, right? It's the organism that you can use in your country, in your environment. It just turns out that in Singapore, you know, the mussel is available readily. You know, it's in the ocean, etc. So there's something that we can use. So in your own setting, what is, you know, available? Something that you can explore, something you can collect in a sustainable way, not to decimate the ecosystem. So yeah, so something that's for you to start exploring first. What was the second question?
Maybe it's available online, or maybe we can download it or something like that.
Yeah, I can send maybe you the link. If you're interested, Prof. Atik can share with the class the link itself. Yeah.
Biology is always develops rapidly, and then
that's where you know, as you can seek help from university lecturers, etc. So I give you an example. One of the more recent program that involved secondary school student at the SST. This is the Science and Technology School in Singapore. So in that program, it's still ongoing. This is our last week was our second meeting. So the start of the meeting was the first meeting was just icebreaker, you know, the scientists from the university meeting the student as well as the teachers, all right, and I get to know each other. And then the first thing we did was to assign a scientific publication, you know, high level, to the student. The objective is for the student as well as the teachers, all right, to convert the science article into a 500-word article for general public, for a newspaper, all right? So part of the exercise will need the student to understand the article, all right? So in order for the student to understand, they have to read up the article first, highlight the words, you know, and etc., and then go online, you know, and search it to understand the paper itself. So the second meeting, the student will come to us again, and then the student will ask questions. You know, oh, why did you do this step? What is this machine? What is this term, etc. All right, and then as scientists, we'll try to explain, and then there'll be a third meeting. In fact, next week is our third meeting with the student again, as well as the teachers. So from that exercise, at the end of it, after 4 weeks, the student's supposed to come up with a text, right, to explain what the article is all about. So the whole reason for doing that is to sort of, one of the objectives is to level up their knowledge, not just at the school, but also what is the current trend in research, you know, and how we as university scientists, how we pitch it to the school level. So these days, you know, it's very easy for us to do science and then just at this level, it doesn't come down, it doesn't translate down. The only time it translates down is when it appears in a textbook. But that can take years, right? And so one of the way to fast track that is interaction with scientists. That's the best way, you know. But of course, you need to engage the scientists, the school, and that the university relationship has to be established. Then, of course, you need to convince the lecturer at the university, because this is something extra for us. You know, we also have our own KPI and research output, etc. So we need to have the buy-in from the university professors, etc., to be engaged, to participate in this program that is worthwhile. So, so I'm not sure if it answers your question, but that's one way of sort of bridging the gap in terms of knowledge, not just in biology, but also in chemistry and also physics, etc. Right? Things are always evolving, as you mentioned. New techniques, new methods of doing things, new advancements in science. So the program that I talked about is not just chemistry, because my background is in chemistry, but it also involves physicists. So they talk about nanotechnology, all right? And these are, you know, postdocs assigning a paper to high school students. Of course, oh, it's all about mathematics. So then the scientists will have to explain, you know, slowly explain, etc. So it takes time, you know, it's not instant. Yeah.
Okay, my name is Suhaiter Hamnam from Computer Education Study Program. I want to ask a question. The question is there a role for computer in the research that doctors do, and what are the rules for computers? Okay.
Your question. It's tremendous. Okay, I'll give you an example of my own research, not science education per se, but my own content research. Is that one of the things that we are now looking at is metadata. Not just a single data, but data with from 1,000 points. So we're looking at trends, you know, in data, and how do we make sense of that massive data, metadata, right? So that's where computational science comes in, where the marriage of data with the help of computational science will try to make sense of the data using or developing new algorithm, for example, right, to process massive data to help us scientists to look at trends. All right, so that's something that's the role of computational science is important, and it's how you can, you know, come up with new program. For example, not just in chemistry but also in biology, bioinformatics, for example, where you have massive data. how you make sense of that. Also in artificial intelligence, right? That's something that is up and coming. So, so all of this is very important. I mean, the role of a computational scientist is extremely important. And this is where we take an integrated approach, not just, you know, biologists or chemists, but also in collaboration with computational scientists to come out to develop a tool that will help us make sense of information. Because nowadays we're dealing with massive data. It's not just one data point, but a million data points. You know, how do we make sense of that?
Oke, boleh tepuk tangan. Semoga yang tepuk tangan bisa menangkap ininya ya, hasil dari diskusi tadi. Mungkin sedikit bikin summarize-nya. Yang pertanyaan pertama dari Bu Lutfi itu berhubungan dengan STEM. Apakah STEM itu harus sains, teknologi, engineering, dan matematik? Nah, ternyata tidak harus se-rigid itu, sehingga yang perlu ditanyakan itu adalah sebenarnya apa sih yang kalian ingin agar siswa kalian capai dalam proses pembelajaran, sehingga nggak harus ada yang proses creating something seperti proses engineering-nya. Dan karena di sini sebenarnya tujuannya adalah untuk melatih sikap ilmiah dari siswa-siswanya. Selain itu juga tadi ditanyakan apa sih peranan dari pendidikan, peranan dari ilmu komputer atau computer science dalam penelitian ini. Maka beliau bilang bahwa ada, yaitu untuk membantu melihat tren dari data-data yang sudah dikumpulkan, sehingga perlu ada upaya kolaborasi antara ilmuwan dan juga data scientist untuk kemudian melakukan mengambil kesimpulan atas apa yang sudah didapatkan. Oke. Maybe I will. Is there any more questions from our audience today? Apakah ada pertanyaan lagi? Okay. I will counting 3. If there are no more questions, maybe we will summarize our general lecture today. 1, 2, 3. All right. I think all questions have been answered. And for the last part of this general lecture, maybe you will be delivering some kind of closing statement, just to wrap up the general lecture session. Yeah, like a closing statement.
Well, yeah. First of all, I just want to thank all of you for attending this, and hopefully, what I presented is useful. Thank you for your time in coming here. I know this is sort of last minute in planning the talk. I do have a second talk, but I think I'm not going to do it because I think everyone is tired. So yeah, maybe next time if you invite me back. So yeah, I mean, I think most of you are going to end up to be teachers. So, you know, in one of the points that I mentioned, there's no limit in learning, right? So maybe that's the take-home message. There's no limit in learning. Try to upgrade yourself constantly and make learning relevant to you as well as a student. Thank you.